Hello and welcome to my Undergrad Research Symposium presentation. My name is Ian Angstrom. I'm in Sullivan Lab with Fred Hutch, and I'm a senior in biochemistry. This project investigates aspartate as a metabolic constraint in cancer cell proliferation. First, I'll give a quick introduction to aspartate and its metabolism. Aspartate is an amino acid that is essential for cancer cell metabolism and proliferation. Cancer cells need lots of nucleotides for DNA, amino acids, and proteins for keeping up with their altered metabolic demand. As you can see in the blue diagram on the left here, aspartate is not only incorporated into proteins, but is also a substrate for nucleotides and other amino acids, notably asparagine. Aspartate is converted to asparagine by asparagine synthetase, or ASNS. While asparagine is used in the production of protein, it does not increase cell proliferation rates like aspartate. Our hypothesis is that ASNS expression and activity can affect aspartate levels, and with this we aim to determine if ASNS expression could alter aspartate availability and change sensitivity to aspartate suppressing therapies. Because cancer cells express ASNS to varying degrees, a secondary goal of ours was to determine if ASNS expression could be used to identify those cancers which are most amenable to aspartate suppression therapies. We wanted to determine whether ASNS expression in cancer cells changed their sensitivity to the complex 1 inhibitor, either rotenone or metformin. These drugs inhibit the electron transport chain, or ETC. Without a functioning ETC, NAD+ cannot be oxidized from NADH, and reactions requiring an NAD+ cofactor, such as the synthesis of aspartate, will be impaired. To test sensitivity to rotenone and metformin, we set up a proliferation assay. We used 143B osteosarcoma cells and infected them with inducible ASNS. Using an inducible system allows for a better control and bypasses the discrepancies in cell growth rates that can arise from single-cell cloning. Here you can see the Western blot for the single-cell clones with induced ASNS. We then added nuclear RFP to the cells so that their nuclei could be detected by an incucyte. An incucyte takes images of multiple areas in each well and automatically tracks growth rate by counting the number of nuclei. When we looked at the incucyte data, we saw that cells expressing induced ASNS had a lower IC50 compared to the cells with wild-type expression of ASNS. The IC50 of induced ASNS was significantly increased when asparagine was added back to the cells. This indicates that ASNS activity can affect aspartate levels, and that it alters aspartate availability as well as changes sensitivity to aspartate suppressing therapies like metformin or rotenone. The next step in this research is to replicate these results across different cell lines. I'd like to give a huge thank you to Dr. Lucas Sullivan and Christian Davidson for their continued help and support with my project. I would also like to thank the Mary Gates Endowment for Students for helping to fund this research, as well as Dr. and Mrs. Levinson for their support through the Levinson Scholarship. And of course, I'd like to thank the rest of the Sullivan Lab for being awesome and being great friends and allies. Thank you.